Hello everybody, thank you for joining today's session. We are in for about 60 minutes of a support session on building models, which is the session 8 in our module 1 of Seismic. I'd like to welcome my co-tutor, is Rob Stanley here. He'll be joining us, looking at your chat messages and maybe showing us a few tricks when simulating ordinary differential equations. Okay, thank you for coming, Rob. I want to introduce the session structure. So we can see it here on the first slide. It gives an overview of the 4 exercises that we're going to work out together, all based on the exercises that are needed for the completion of assignment 8A8, the assignment that asks you to model the blood glucose level based on some surprising recent results that actually to control the blood glucose, insulin is not necessarily required. Insulin also does that job, but there's other potential inhibitors in mammals and potentially also in the human. So we're going to take a look at something very similar. However, I'm going to work with a different model today to enhance also your repertoire of those things from the literature that you are familiar with. First exercise will be to look at a nonlinear function that the model uses. You remember nonlinearity is a key feature in all models because it allows us to get more surprising, more interesting, more complex solutions than from the linear results. And in general, we think that all of biology is based on the existence of nonlinearity in the interactions. We'll then look at an algebraic way to find steady-state solutions or a special feature of our model. And we'll continue with looking at the so-called symbolic solution of a model, a way to manipulate mathematical expressions symbolically, as it says, and rearrange them conveniently and give us some information that is of general interest. And finally, we'll do some numerical simulations of the model to see some features which we cannot really pick up by the analytical solutions that we get in the previous functions. So there will be additional things we get out of the simulation that we do in the end. MATLAB code is available for you. And for the following, I think you have now seen me and Rob. Please allow me to switch off my web camera. And also interrupt here. Right, let's get into the session by looking at the theme that we are dealing with. Our theme is actually some of great biological interest, namely the role of control loops. Control in the sense of feedback, where some part of the system, of your biological system, some molecules, some membranes, some cell, some entity, has an impact in its current state on other members of the system, means other molecules, other compartments, other cells, other tissues, by ways of interacting with them. And we can see in that scheme that we have depicted here on our second slide, an example from signaling pathways, that typically in biology we're not dealing with a single feedback loop, but we have multiple feedbacks involved. This makes the prediction of what's going to happen to a system or to a biological experiment, if I interfere with it, very, very difficult, if not impossible. So what we are going to do now is we learn how to set up a model in order to investigate what features might be expected from certain types of control loops. And if you look at the paper from which this graph is taken, you can see that the modern topic of today is to study the interactions of multiple feedback loops. Whereas today we are going to start simply by looking at the impact that a single feedback loop has. Nevertheless, its impact will be very important for understanding what we are doing, also for predicting potential outcome of experiments. So the model that I chose is a variant of the one that's shown in this third slide here. Brian Goodwin, who used to work at the Open University in the United Kingdom, developed that. He's, I think, original Canadian, and it's extensively studied in his book from 1963 on the temporal organization of cells.
Could we perhaps make the image bigger?
Okay, I can make this a little bigger. So it's maybe easier for you to see. So we have the equations that we're going to look at in more detail later in the yellow box, and some scheme that is supposed to tell you that what we are looking at here is a cascade of 3 consecutive reactions, one with molecule X at its center, one with molecule Y at its center, one with molecule Z at its center, where the last of this cascade, namely molecule Z, exerts a feedback on the production of the first molecule in this case. And what Brian Goodwin then said is that the most important feature that has been found in, for example, in metabolism, in signaling pathways, and in secondary synthetic pathways, amino acid synthesis, and so on, is that there is negative feedback, such that the product of the cascade regulates what happens at the entrance of the cascade in a negative way. And of course, the main idea is simple. You want to have an interaction that models. If I have too little of the product, I want the cascade to work fast. I want a lot to go through the cascade in order to produce more. If I have too much of the product accumulated, I want to have the cascade stopped or slowed down at least for a moment in order to get rid of the accumulated product. And as such, negative feedback plays an important role. It comes from control theory, basically, but it's found in abundance in biological systems. And let me just advance here.
Zoom back out.
Just a small second. Going to the final slide. We are looking at this scheme in a simplified way. So what we have here is the actual equations that we are going to look at. So the first thing is to look at our yellow box here. And we can see that we have simplified our cascade by considering only 2 types of molecules, rather than 3, and as in the original Gibbs, we have simplified it so you have the same number of variables as you have dealt with in your first simple EPO model, or the same number of variables that you're going to use in your physiological model for the assignment. So if we look at the differential equation, it shows us that both variables in their rate equation have 2 terms, one positive, one negative. 3 of the terms are very simple. They are just simple variables. 1, 2, 3. 3 are just simple linear variables, and they don't even have a rate constant, means that in this case the rate constant is set equal to 1. So if I just said this way, it's an easy modeling of a kinetic cascade where the output of 1, x1, goes into the output of the second x2. Now the feedback is now expressed in a complicated nonlinear function, so the production of x depends on something that is governed by a parameter a, a constant, divided by the sum of another parameter, km1, plus a function of the second variable called x2. If we set the amount of x2 equal to 0, or if the constant that governs this inhibition, which is called ki, either of this is 0, then nothing will happen in terms of feedback control. But if some species x2 is present and the parameter ki is not equal to 0, then we'll have a situation where the amount of x2, the second species, will actually inhibit the production of the first one. And I say inhibit because what we have here is that it's in the denominator. If you increase the amount of x2, then dividing a by this number will make a smaller number. If you divide something by something that gets larger, you will get some smaller number. Means the input into the molecule x1 or the concentration of x1 will be diminished. And this is our way of expressing feedback inhibitions. For the present, I have chosen a certain set of parameters. And if we have some time at the end, I might give a little comment on how these parameters are chosen and what the impact of having different parameters might be. For our convenience, we have now written the equations here, also for your convenience, such that we can actually copy and paste the equations and use them in our MATLAB code. And we have them for the algebraic manipulation, and we also have them for the symbolic manipulation